And I have a great pleasure to introduce our speaker today. He's Yair Shemesh. He's a staff scientist, tenured staff scientist at the Department of Neurobiology at Weizmann Institute of Science in the group led by Professor Alon Hen, who is also the president of Weizmann Institute. And Yair has a quite interesting background. He did his master's thesis in plasticity in bacteria. Then for PhD studies moved on to honeybees and then completed a doctoral thesis on plasticity of circadian rhythms in honeybees under supervision of Guy Bloch. And then for postdoc, he moved to the laboratory of Alon Hen and Elad Schneidman at Weizmann Institute, and where he started studies on mouse social behavior, also by using ethological approaches. And this required new monitoring systems, new tools to develop. And I think the first achievement was published in 2013 in eLife, where the system that is tracking behavior of individual mice in social group was published. And today, Yair is going to tell us how rodent neuroethology could revolutionize translational psychiatry. So thank you, Yair, and the screen is yours.
Okay, thank you very much, Vootele, for this nice introduction and the opportunity to present our work in this very interesting webinar series. And thank you, everybody, for joining us. And I'm a research associate in the lab of Professor Alon Hen, and in the lab, we're interested in mechanisms underlying stress and stress-related psychopathologies. And I will talk today about the challenges of translating studies from animal models to humans, and how semi-natural setups like the one that we established, the social box, can help us to bridge this translational gap. So, for some reason, I cannot, okay. Okay, so we're all exposed to stressors. Every organism exposed to stressors continuously, and the brain receives stressoric input that it can be chronic, it can be acoustic, and then it produces the stress response to cope with those stressors. So it is an adaptive. It is an adaptive response. And based on our individual differences, such as past experience, genetic background, epigenetics, the brain produces behavioral and physiological response to maintain stability. What does it mean to maintain stability? It means to provide the body with what it needs to survive. So it is shelter and to avoid the danger, food, water, and mates. So we have this constant environmental changes, and this and the brain that tries to maintain stability through change. And this is what allostasis is about. Allostatic load is the price that we have to pay in order to maintain stability and to adapt. When the price is too high, the response might be maladaptive, and then it might develop into psychiatric disorders. And psychiatric disorders are a serious issue. It has a very heavy toll on individuals, on family, on society in general, and we would like to treat them. In order to treat psychiatric disorders, we need to diagnose them. Psychiatric disorders are diagnosed not as any other diseases based on biological markers, but based on the symptoms, on categorization of the symptoms, like the DSM-5, for example. And this poses some issues. One of them is comorbidity. You might have different disorders that share the same symptoms, and also heterogeneity. So the two different individuals might have the same disorder, but still have different symptoms. So you can imagine that if the diagnosis is complicated, then finding the mechanisms that underlying those disorders in animal models and translating them to humans, it's even much harder. And it is especially hard because the behavioral readout that we extract today, and it is our ultimate actually output, the behavioral readout. This is the symptoms in humans that we use to diagnose the mental disorder. So in mice, the behavioral readouts are too simple and they lack validity. And in order to understand why we have this problem of validity of the behavioral readout that we extract from the classical test, we need to talk about the ways to measure behavior in the two disciplines that were developed early in the 20th century, behaviorism and ethology. So behaviorism is basically based on short tests in the lab using animal models, mainly rodents. And the main advantage of these tests is their high controllability. And that you can repeat those tests quite easily. They are simple, using many animals. But of course, those tests fail to detect longer-term behavioral patterns, and they lack the natural complexity. So the readout itself might not be relevant to the natural behavior of the animals. A very classical test that we all know is the open field, for example. It's basically an empty box. We take the animal, we put it inside the box, and the time that the animal spends close to the walls, as the relative to close to the middle of the box, and if it spends more time close to the walls than compared to controls, then we would say that the animals tend to be more anxious. We try to be careful and call it anxiety-like behavior. Anxiety involves many emotions, and emotions are subjective. We cannot really ask the animal about its subjective feelings, but it is a response. So it is response. We can actually quantify the response to this anxiety state. But again, those tests are relatively short, and what they do lack is the adaptive value of the behavior. We don't really know if this behavior is less adaptive or more adaptive when we study a specific manipulation that we do. Also, when we study social behavior, for example, the three-chamber test, most of the interactions are between dyads. Again, it's short tests. The animal are we have an actor, and then it interacts with a restricted animal. The animal that is restricted behind the mesh, and we measure the time that it spends close to the restricted animals or in the empty chamber. Now, what we miss in this setups is actually the social context of the group, and we know we treat all the animals as similar. They are either controls or treatment animals, but they are all within the treatment are similar, and within the control are similar. But we know that when you look at the group, there is hierarchies, and those animals do not behave similarly. They are individuals, and we just ignored that. And one more thing that we have to keep in mind, that there's also the experimenter effect. We take the cage from the rack, and we take the animals from the cage, introduced it into the apparatus. This all introduces stress to the animals, and it is also something that is hard to control. So those, and there are also many other tests that I will not get into now, but you understand the concept. I'm sure that you're also familiar with it. Ethology, on the other hand, was basically developed to study the behavior of the animals in their natural habitat. And it is focused on innate behavior and how those innate behavior relates to survival, how it increased or decreased the fitness of the animal when it gets rewards or danger, or as Nicholas Tinbergen put it, interviewing an animal in its own language. So of course, the behavior is much more elaborate, But there is one very important issue. It is we cannot study the mechanism, the molecular, the neuronal mechanisms that underlying behavior in the natural habitat. We can manipulate the environment to some extent, like the famous von Frisch experiments with the honeybees, but we cannot really use mouse genetic tools to manipulate the brain, to record from the brain, to quantify gene expression, and so on. Now, semi-natural setups in the lab are actually can take the best out of those two approaches and that were developed totally separately during the last 100 years and try to combine manipulations together with complex behavior. And Robert Blanchard is considered the pioneer of semi-natural setup. There were other groups that also before him worked with semi-natural setup, but the visible Borer system was, he dedicated his life to work with the visible Borer system, and we were very much inspired from his work. So the visible Borer system is basically can be, you can work with it with mice or rats. It's an open surface. It is quite a big setup. Yeah, it's quite a big setup. It's 1 square meter, and we have an open surface and also tunnels that connects to chambers with water and food. And back then, of course, there was no way to track the animals automatically. So he used skilled observers that observe the behavior either live and wrote what they see, or then later on also he recorded the behavior using videos, specific time points, of course, because the animals were kept in this visible forest system for days and even weeks. So of course it is not feasible to observe the behavior all through this time. But at specific time points, there were observers that observed the behavior, and he reached very interesting insights about the importance of the group, about the importance of the structure of the group, the size of the group, females and males in the group, aggression, stress, and so on. So as I mentioned, we were very much inspired from this work. And when I joined the lab as a postdoc 15 years ago, we decided to build a semi-natural setup, but this time that the behavior will be recorded automatically. So this version that you just see in the picture is actually a recent version. It is we worked with I will show you videos with recordings of of the first versions that we use, but the concept is the same. We have groups of animals that are kept here in this in this arena. It's 60 on 60 centimeters. We mostly work with groups of 4 animals, and there is an enriched. It is an enriched setup, so there is a nest and also other objects and drums that they can take shelter and so on. And there is, of course, bedding. They can forage for food. The food can be supplied either ad libitum or at specific time points automatically. And we color mark the fur of the animals. We use dyes that remains on the fur for many days and even weeks. And we prefer to work with albino animals because the dyes holds on these albinos animals much better than on black mice. So we work with ICRs, and if we and ICRs are also outbred. So it is also an advantage because their behavior is more elaborate. There is more aggression, more social behavior in general, and also the findings can be relevant for many for other lines of mice, and it is not specific for specific line when you work with outbred. So if we need to work with transgenic animals, we cross them with the ICRs, and after 2 generations, we have mice that are albinos, some of the mice, of course, and then we can establish a family. We, of course, track the animals using a very sensitive light camera under 1 lux. So it's a very low light, almost a moonlight. And this is how it looks like. So this is the old version of the social box. Most of the papers that we published so far are either with this old version or also with the old tracking system. And you can see here the animals, the different with different colors. And at this point, when we had this video, we subjected it to automatic tracking using a MATLAB code. Here I collaborated with my friend Oren Forkosh that wrote this code. And the idea is that we have 24 frames per second. So for each and every frame, we differentiate the colors from the background and the specific colors one from another. So we can actually automatically detect for many days the locations of the animals in the arenas, their coordinates. And based on that, on this vector of locations, we can also extract other complex behaviors. So as I said, most of our studies so far, we used this code, but in recent years, there are more and more platforms that use deep neuronal networks for pose estimation of the animals. So the idea is that you have to train the network on, it's a supervised approach. So it's supervised machine learning approach. So you train the network on few hundreds of frames, preferentially also, or some of them should include complex behaviors like attacks or groomings and so on. And during this training, you actually mark the different body parts of each and every animals and each and every color, and the system learns to recognize it. And then you run a tracking on the video, and it is extremely accurate. This is the deep LabCat. This is the one that we use, and you can see how accurate it recognizes each and every body part. The last version of the deep LabCat also has a multi-animal, it calls a multi-animal deep LabCat. So when we use the multi-animal deep LabCat with our labeled mice, because we still have the color, even if the animals gets into the nest and disappear, it is recognized once it is reappear, and there is no confusion between the animals. It's really working great, and it gives us a whole new opportunities to measure behavior, because now we have 8 body parts of each and every animal. So we can easily detect, for example, unogenital sniffing or face-to-face sniffing. And then we can take the data to other platform. For example, Simba, that works on, that it's another machine learning based platform that is based on random forest. And we can create libraries of different behaviors. For example, of attacks, we can, using skilled observers, collect those behaviors, train Simba on those behaviors. And because we have such an accurate pose estimation, we can detect complex behaviors like attacks or self-grooming automatically, something that we couldn't do before based on our old code. So this is basically the capabilities of the social box. And in the last slide of the talk, I will talk about a setup that we established that has 16 arenas with 16 groups that can be run simultaneously. But this is how we started. And if we conclude this theoretical part maybe of the talk, we can actually think about many ways to measure behavior. And by no means I suggest that semi-natural setups are the only or the best way to track animals. And I can show you studies that we actually combined more than one approach in order to get to a full picture. And I think that this is the way to go. And if we can think about the way to measure behavior in 3 axes, so you can think about, of course, and there are pros and cons for each approach. So you can think about like the x-axis, it's the environmental complexity. So the environmental complexity can be either standout, very easy to, or relatively easy to perform, like a test of the 3-chamber that I talked about, or the resident intruder. Also home cage monitoring, you monitor the behavior in the home cage itself. And then you can have setups that work on enriched environment, like the visible border system or the social box. And there are different levels of enrichment. And then you can have a natural environment. For example, ethology, which is also important. And actually, I think that there is really lack of studies on mice behavior in nature. And there is very interesting setup that is named the etholoop. It was published a few years ago, which is much more ethologically relevant or natural than, for example, the social box or the visible porous system. It's just that establishing it is very, very complex, and you have to use a few cameras and also a large room. So this is the environmental complexity axis, if you like. And then there is the tracking duration. And again, there are pros and cons. So you have short tests that they can again easily be done with quite a lot of animals. And I remind you that when you work with the social box or the visible border system, then the sample size of a group is 1. So you have to run many, many groups, and the sample size in those tests is actually a single mouse. So those are short tests. The home cage monitoring is continuous and prolonged, but you get the transition of the animals from one area to another. So the behavior is fragmented. You don't get the whole behavior, and then you have a prolonged and continuous tracking of the behavior each and every frame, and you can extract many behaviors like in the social box or the etholoop. And of course, the complexity of the behavior, it's like that can be regarded as maybe the z-axis, and it can be simple interaction between a mesh, or it can be complex, like in the resident intruder and visible border system, ethology, and the social box, that you get more complex behavior, chasing, sniffing, self-grooming, and here either you observe this behavior, or you need to establish libraries and train a model on those behaviors. So let's talk about some studies that we done using the social box, and as Vootele mentioned, that was the first study that we published in 2013 with the box. And this study was based only on the location of the animals in different parts of the arena. We divided the arena to 10 different zones, and we just took the vector of the locations of each and every mouse out of the mouse, or out of groups of 4 in those specific zones. And we ask a very simple question. We ask how much those animals are independent one from another, or how much they depend, their location depends on the locations of the other. So we have 10 zones, we have 4 animals. There are like 10,000 different configurations that those animals can be in. And we can look at each and every animal on the places that it chose to be in the arena, and calculate the probability of this animal to be in this area. And then we can calculate, based on these probabilities, the probability to get a specific configuration, that these two animals will be on the box, this one on the feeder, and this one in the open area. And when we did that, when we calculated the probability of the configurations based on the individual probabilities, we saw that we couldn't predict well the distribution of the configuration, which means that there is more to those configurations than just a single behavior. The animals are not individual. We use maximum entropy approach, and we included now dyadic interactions or triple interactions. And when we did that, we see that the predictions becomes better and better. The animal use also information on the location of the other animals in order to decide where to be. So the group is more than the sum of its individuals. And this information might be very important in many studies. But we have more than just the locations of the animals. We can extract from just the center of the body. And again, most of the studies that we did so far were based on the center of the body and not on the data from the deep lab cut. We can extract velocity and distance and acceleration, angular velocity, many parameters. We can also extract parameters that might be relevant for anxiety, like the distance from the wall, exploration, and so on. But we also calculate dyadic interactions. we can base on the angle of one animal to the other animal and the distance between them, and whether the animal moved towards the other animal or away from it, we can extract behaviors like approach, avoid. If one animal approach the other and the other animal avoid on the same time, then we can see a chase or a follow behavior. And then based on those chases and follow behaviors, we can actually extract hierarchies. Who chased whom? So there's so much information that we can extract just based on this simple center of body of the mouse, and those parameters are actually individual parameters. They differ between one another. Of course, the dominant will chase more, the subordinate will chase less. So now we have a tool to quantify individuality, or if you like, personality. And if you talk about personality, then the definition of personality is the existence of behavioral differences among individuals. So you need differences between individuals of the same species, which are stable over time. And of course, different contexts and situations. So we took this definition, and of course, as I just mentioned, we have many readouts. We have tens of different readouts. So we will need to use a dimensional reduction approach. But let's now, in order to understand what we did, let's talk about two readouts, chases and exploration. And imagine that we have two animals. One is blue and the other is orange. And we have data from 4 different days for the blue and for the orange. So basically what we look is for an axis that will minimize the difference within the animal and maximize the difference between the animal. And this will actually answer the definition of individuality. And for that aim, we use the linear discriminant analysis for dimensional reduction. And if we choose this axis, and of course, it's the same concept if you use many dimensions, we try to find the minimal mouse variability within and the maximal between. So that's the concept. And we asked, okay, do really those? Of course, there are differences. You just look at the videos, and you can see that the animals behave differently. But is it just because the current environment, or is something innate in their behavior? Those individual differences are innate to the animals. So, in order to ask that, we first did something that is extremely extreme interference. We reshuffled, we took 4 groups of 4 animals each, and we reshuffled the group. So now you have new hierarchies, and of course, it's a very stressoric situation. And then we asked, okay, how the dimension, or we call it IDs, which is basically parallel to PC in PCA. So how the IDs will correlate before the mix and after the mix. If we find correlation between those IDs, it means that something remained even after such an extreme manipulation, and that there is something innate in those individual differences that we see. And indeed, we saw significant correlations in the first 4 IDs. And then we ask something more, because if you remember, one of the definitions is that those differences have to be stable over time. So we took animals, we took groups that were 4 weeks old, introduced them to the social box for 4 days, and then brought them back to their cage, and then at the age of 12 weeks, introduced them again. And again, we did this correlation between the IDs during when they were juvenile and they were adult. And again, we find significant correlations in the first 4 PCs. So we actually have here a tool for quantifying predispositions for different manipulation. And imagine that we can now quantify individual differences before a very stressoric manipulation. For example, a PTSD in the social box, and then look at those predispositions of and how they might affect the susceptibility to the manipulation that we're doing, and it can be a very strong tool. Okay, so remember that I told you that it is important to combine different approaches, not do all the experiment only on the social boxes. And this is one study that actually showed that. Here we worked on urocortin 3. Urocortin 3 is a stress system component and highly expressed in the medial amygdala. The medial amygdala is a very social brain area. And we suspected that it is involved in social stress. So before doing very complex manipulation in the social box, we did a simple experiment, not that simple, but relatively simple, optogenetic activation of the urocortin 3 neurons in the medial amygdala while interacting with other animal through a mesh. So you can see the optic fiber here. We activate the urocortin 3 cells in the medial amygdala. Of course, we express the channel rhodopsin in the urocortin 3 crystalline in order to achieve that. And what we see is that following light on, the mice tends to spend more time following activation with a novel animal and less time with a familiar one. So it increased the interest in a novel animal. We don't see it in a light off, and also we don't see when we don't see it when we block the access to the of urocortin 3 to the receptor with an antagonist just before the manipulation. But now we asked ourselves, okay, but what does it really mean? Because we just see increase in social interest. It can be agonistic behavior. It can be a pro-social behavior. So we decided to check that, and this time we use chemogenetics in the social box. And in the social box, those groups are all familiar. So it was with familiar, and we inhibited this time the urocortin 3 cells. So since we inhibited the urocortin 3 cells, we expected to see this time elevation in the interest between those familiar mouse. And we said, okay, this is here we can actually look at the time that they spend outside the nest, because the dominants keep pushing the subordinates into the nest. So we can look at the time that they spend outside the nest, or the number of approaches one towards another while they are outside the nest, and learn whether it is a pro-social behavior or aggression. And we saw that when we inhibit the urocortin 3 cells in the medial amygdala, we see an increase in pro-social behavior. The animal spends more time outside the nest. They were more socially tolerant one to another, and also interacted more and approached each other more. And this is a nice example how a combination of different approaches can give us new insights. One advantage of working with semi-natural setups is the ability to track the behavior for prolonged time period. And with oxytocin is a very good example why it is important. Because we know oxytocin as a pro-social agent. We know that it increases affiliative behavior, cooperation, it even attenuates stress. But there is more and more evidence that oxytocin can also increase envy or schadenfreude and even defensive aggression. When we work with animal models, we're doing short manipulations, short interactions, and we don't see increase in aggression in the resident intruder test, for example. But what happens if we activate the oxytocin cell for prolonged time periods? Maybe we will start seeing more complex interactions when we will do that. So in order to achieve that, we wanted to activate the oxytocin neurons in the PVN using optogenetics and not chemogenetics, because optogenetics is much more accurate in terms of temporal resolution. So we developed a wireless optogenetic device in order to achieve that. And this wireless optogenetic device can be activated automatically when the camera recognizes a specific mouse by activating an electromagnet just above the feeders or any other place that you decide to put it. Is a very simple device. It took us a lot of time to develop it. It costs 1 gram. It weighs 1 gram. And so it's relatively not heavy. And we used a mutated channel rhodopsin that is named SSFO. So SSFO, upon short pulse of light, you don't have to have a long activation, just 3 seconds of light, it causes a sub-threshold desensitization of the neurons. So it is more physiologically relevant, and this sub-threshold is for half an hour. So we can give every 2 hours automatically active when the animals is in the feeder. So it's approximately every 2 hours we can activate this device, and this is how it looks like in the box. The animal reach, and you can see that the light turns on, and now there is a sub-threshold deactivation, desensitization of the neurons, and so it is more sensitive. And now we ask what happens after 2 days of activation. And it is important to mention here that there were few experiments that tried to activate the oxytocinergic system for a prolonged time period, but they used mini pumps and pharmacological delivery of oxytocin. And this caused desensitization of the receptors. So you have less receptors now because you deliver a lot of oxytocin, and then the interpretation becomes very complicated. When we use this very delicate manipulation in the social box, we did not see any desensitization of the receptor. So in the first day of activation, what we actually saw is increase in prosocial behavior. As expected, we saw more contacts in the CRE plus animals, the ones that actually express the oxytocin. They spent less time alone outside, they were more social. What happens on the second day? On the second day, we saw something different. We saw that this increase in prosocial behavior also increased agonistic behavior. They chased each other more. They did more anogenital sniffing. So we can see that prolonged monitoring of behavior while doing specific manipulation, very delicate manipulation actually, in the social boxes is possible, and it also delivers very interesting insights. So the last experiment that I want to talk about is when I started this talk, I said that I mentioned that when you track animals in a semi-natural setup, you can actually learn about the adaptive value of the behavior. And this is exactly the idea of ethology. Ethologists that study behavior in the natural habitat, they look at innate behavior and how they increase or decrease fitness. And we decided to do it with aggression. And aggression is interesting because aggression is an adaptive behavior, right? We must be aggressive in order to gain resources. There is competition of the resources, but too much aggression is not adaptive at all. It is dangerous, and it is also costly energetically. So there are social dominance hierarchies. Agreed differential access to resources that attenuates aggression. So we have this equilibrium and we have this threshold, and how it is decided and when this threshold is not adaptive anymore. That was the question that we wanted to answer. This is an unpublished data. And we decided to do it in the social box because in the social box, we can actually look at aggression and social dominance hierarchies together. This is something that cannot be done in classical tests. And we had 2 setups. One, we had enriched environment from adulthood, from weaning. So during adulthood, the animals were grown in an enriched environment, very super enrichment, very crowded. After 6 weeks, we color marked the animals, only 4 of them. We excluded the rest and then put them in the social boxes. Enrichment increased cognition, but it also increased competition and aggression. So this is an adaptive model. But we also works with oxytocin receptor knockouts, which when it's a genetic knockout, we know that there is an increase in aggression in this knockout. But we assume that this, because it's not shaped by the environmental needs, will be a maladaptive aggression. So we started with looking at the etograms, and I will show you just part of it because we're running out of time. But we see that already with just looking at the etograms, at the locations of the animals in the different areas of in the arena, there is a difference between the standard conditions, the controls, and the environmental enrichment. They tend to be less, they explore less, to be more in the nest. We see exactly the opposite when it comes to the OTR knockout. They actually tends to be more outside the nest, explore more, do more exploration. Those knockouts when they are in a group in the social boxes. We also see it more in the interactions themselves. So then we ask, okay, what about aggression? And we looked at the total number of aggressive chases in the boxes, and we saw something actually that seems to be counterintuitive. In the environmental enrichment, we saw that in total, there is less aggression compared to the standard condition. In the OTR knockout, there is much more compared to their control mice. But then, when we looked at the contact rate, we saw that in the environmental enrichment group, they are doing much less contact. And if you normalize the number of aggression to the contacts, then you see that for each and every contact, the probability of it to end in aggression is much higher. So they actually use two strategies. One is social avoidance, reducing the total number of aggression, but once there is a contact, then the probability that it will end in aggression is much higher, and this establishes a hierarchy. And when we look at the oxytocin receptor knockouts, we saw something different. We saw that they're actually doing more contacts, and when we normalize the number of aggression to the number of contacts, there was no difference from the control. So they're just interacting more, chasing more, there is more aggression, they waste more energy. And I will leave you with this part in this project. There are many other findings in this project, but we will publish it soon, and you will have to wait to hear about the differences in hierarchies and also in the competition over the resources between those animals. And I want to sum up or with actually talk about what's next. And we're nowadays establishing, because remember that the sample size here is n equals 1. So we really have to find a way to enlarge the sample size, and we build right now, and it will be ready in a few months, a setup that will include 16 arenas that can be run simultaneously. And not only that, each 4 arenas will be connected one to another, be recorded by one camera. So we can actually learn about territorial interactions between different groups of the animals. So it is kind of an intergroup interactions as well as intra-group interactions. And it will be relatively easy to establish with the together with the pipeline of recording the behavior and also monitoring it simultaneously with deep lab cut. But more than that, we would like to induce challenges in the environment. We saw that if the environment doesn't really change much, many behaviors just drops after the second day. They just learn that there's nothing to do in the arenas, and they just stay in the nest and go out to eat. So we develop or introduce into arena automatic ways to challenge the animals. For example, if you can see here, this tower, they need to get, in order to get food, the food will not be available ad libitum. They will have to climb in, stand on this wire, and find a way to get to this food in this automatic feeder. But once in a while, they will get a signal, and they will have to run away on time in order to avoid getting a food shock. And this is all will be automatic, based on recognition by the camera and using Arduino that will activate all these complex situations. So now there is a danger and there is a need to forage for food, and we can study about their threat response and really get a very accurate way to measure maladaptive behavior once interacting in the social box. So I will finish with that, and I hope that I convinced you that semi-natural setups in the lab are relatively easy to work with, and that they can gain us with a lot of new insight about complex behavior. So again, I want to thank Vootele for inviting me, and I want to thank Alon Khan, the head of the lab, and all the social team, great people that work with me throughout all the time, and thank you for listening.